And so yeah, and so that's kind of where the idea of Polo came out, and ultimately was the goal of the Polo program. And this was we published this in a short paper in 2021 that you can check out if you're interested. And so I guess a little background on the development of Polo. This was done through the BioXFEL summer internship, which is a really great program. I would definitely recommend it to your undergrads if you have them. I was mentored by Sarah here, and it was 10 weeks of fully CDC compliant Python programming to produce Polo. And so yeah, I had a lot of fun doing that. But a little bit more about the actual program and some of what I think are the great parts of Polo and why you might want to use it. So this kind of overview image is showing you the main kind of graphical interface that you would probably interact with in Polo. And kind of like, I've been talking about the key feature here is the automated image classification combined with this visual interface. And so some of the things you can see, this is called what I call the slideshow interface, because you're generally looking at one main image at a time, shown here in the center. Can you see my mouse? I just want to make sure I'm actually pointing at something.
Yep.
Okay, cool. So here we're looking at one example image in the visible spectrum. And you can see in some of the other areas, we have details about the image, what well it's from. We see that these images have already been classified by the Marco model, and it's marked them as crystals, which makes sense. Glad it got that one. And it's very confident about this. So you can also see the confidence score for whatever automatic classification. And then you can also see the cocktail details that are linked to this specific well number. So that's all available in this main interface. And then over in this section, which I think can also be really useful, is oftentimes your one sample is it's going to be imaged over a time period, and you want to be able to see the progression of crystal growth if you have all of those sets of images. And so you can hit this checkmark box here to show if you have any additional runs that are of the same sample but taken on a different day. Polo will automatically kind of collate all of those images and produce kind of this labeled timeline in this interface. And you can also swap to any other spectrums that your image in, such as we talked a bit more about the UV TPEF spectrum done at the crystallization center to really easily verify that the image you're looking at is in fact it looks at protein versus salt crystals without having to open up another tab in your image browser or something like that. So we'll see if this plays. I've got a little video here of that I did a while ago of me classifying some images and actually using the interface. So this is an unclassified run. I'm going to click classify images, and so you can see progress bar starts going. So the Marco model is running in the background, and you can see that the image classifications are kind of starting to come in our current run. And so I think here's we're just speeding through the classification process because it can take a couple minutes to run because there are a lot of images to look at. And then I'm going to skip ahead for a second. Once you've classified all of your images, you can then apply these different filters. So maybe you only want to see, the most obvious would be just images that contain crystals that were classified by the Marco model. You could also, if you wanted to see images that you classified as crystal or something else, you can also do that once you've gone through and looked at all the images. And that's what you would be clicking on in kind of this classification interface to add human labels to each of these images. And I think I'm going to submit the filters here in a second. And I'm just talking about all this stuff. And you can also change how the images are returned. So we're sorting them by the most confident to the least confident images. And so we think based on that classification, Marco thinks these are crystals. I don't know if they actually are. But again, this might be an opportunity to look at your UV-TPF spectrum to see if those are salts or what they might actually be, because again, the model is not perfect, and so it's always good, you're going to need to go back and look at the ones that were classified as crystals just to verify that, right? And so that's what we're doing here. Okay, let's go on to the next slide. There's also other ways that you can look at your image data in Polo. So instead of looking at one single image at a time, like in the slideshow viewer, you can view them as sort of like a plate. So I'm calling this the plate viewer here. And it's kind of similar to the filtering that we did with the slideshow view. You can select a specific classification that you are interested in. It's usually crystals. And have the program highlight those images that have been classified, that classification has been applied to either by you or the Marco model in a specific color. So here we're highlighting all of the images that Marco classified as crystal with a confidence greater than, I think, 60% or something around there. And then if you see one in this view that you think looks really interesting, you can just click on it and it will kind of blow it up into this mini slideshow viewer where you can apply your own classifications and also look at the details for that image, like what well it was in, the cocktail conditions, and that kind of thing. And you can also visualize where you are in the plate if you're interested in that as well. And this is kind of the other main interface, which is just the table viewer. And this kind of goes along with the idea of Polo as a tool for organizing your image data. So once you've done your image classifications or run your models or whatever you want to do with it, you can also just view everything as basically like you would in Excel. And so all of this data is linked to your actual image data in the Polo interface. And I think the other main feature is that in addition to this classification and run organization, you can also really easily export your data to formats that will be useful for if you have any other analysis programs that you want to run on your classification confidences or something like that. You can export all of that data to CSV or JSON files. So some really easily parsable machine formats. Then you can also export your imaging data to PowerPoint presentations. So if you have a number of wells that look good or have some crystals in that, and you want to share that with, your friends, your cats, collaborators, whatever, you can select those images. And Polo will automatically generate these kinds of PowerPoint slides for you, which have your main picture of your well here, imaged at the latest time point, and your crystallization conditions for that well. And then if you have this kind of other imaging data in other spectrums, it will make a slide for that, as well as creating this kind of timeline type image so you can see the crystal growth in that well over time. And finally, I just wanted to highlight a few other kind of miscellaneous features that are included in the program but may not be used as often. Polo actually has an integrated FTP interface, so you can use this in lieu of programs like FileZilla to download your data from the HWI FTP server. And so Polo will automatically, you put in your credentials, Polo will download that data and then automatically import it into the interface. So it just makes things a little bit easier. And then you can also, Polo will also automatically generate some basic, plots of, for each run, things like what proportion of your samples were crystals, or and which one, how many had precipitate, and how do the human classifications, so the classifications that you're actually doing, differ from the automatic classifications. There's also some like housekeeping things like Polo will automatically back up your human/machine classifications as you use the program in case something crashes, won't be lost. You can add custom cocktail formulations, and there's other miscellaneous features as well. I won't go into detail about. And next, I wanted to highlight some existing resources because I do get some questions about the program. And sometimes I think people don't know about these. There is an online documentation page. I think that's linked to on the GitHub. I think maybe we need to make that link a little bit more visible. But this has a lot of good information about installing the program, frequently asked questions, some guides. And also, if you're interested in modifying the program or changing the code at all, there's also, I would say, relatively decent API-style documentation available through this website as well. There's also some tutorial videos that I put together that just, if you want to watch me use the program and learn about some of its interfaces and how it works, those are linked to on the online documentation, and they're just hosted on YouTube. And then also there's the Polo GitHub page. And if you have a suggestion or you run into an issue, please report it or let me know, send me an email, because oftentimes I don't really hear much about this program. And I'd love to hear how people are using it, what's going wrong, especially why people aren't using it, if they ran into issues trying to figure something out and gave up because the program was too hard to use. I'm interested in all of that kind of stuff. So please let me know through either the GitHub page or you can just email me. And I guess if you want to go to these sites, screenshot now, there's links to the documentation, the videos, and the GitHub page here. Finally, I'm also working on some new updates for Polo that should be coming soon-ish. The main ones being there's going to be releases for newer Mac operating systems. So hopefully that will help if people had with issues installing the program on their Mac machines. I think Sarah alluded to there's going to be an updated cocktail formulation. And so when that rolls out at the screening center, that will also be included in the latest release of Polo, and probably some other minor bug fixes. And kind of related to that is I think there'll be some questions about Polo for both people that have used the program, tried to use the program, have not used the program. But if you could, it would really be helpful if you could give any feedback that you have about the programs, because we put it out 2 years ago, and honestly, I get the occasional email about it, but other than that, I don't really hear too much about if people are using it, what they're doing with it, if it's helpful, if it's not. So all that kind of information and data will be really useful, I think, in the future, figuring out where we're going with this particular program. So that's all I really wanted to talk about today, and I just wanted to say thank you to everyone at the Crystallization Center, especially Sarah. Also, I wanted to thank Bill Bauer from the BioXFL Summer Internship Program. He does a really great job running that program, and it's really where I got my start with this program. And then I also wanted to thank Lisa Keith at IMCACAT at Argonne National Labs. It was an internship with her where I really started fooling around with this kind of crystallization stuff, and she was really instrumental in getting this whole thing kind of rolling. So with that, happy to take any questions, and thank you for your time.
All right, thank you, Ethan. This is fantastic. I'm just going to remind people in the audience, you can type questions into the chat at any point, and comments and so on. But since I'm a computing person, I'm going to jump in with a question first. And so, first of all, thank you, Ethan, it's a fantastic tool, and it really integrates a lot of functionality. And I think, it's really worth it for people to kind of put this together, or put time into getting it to be able to use it. But I was wondering, I'm on the kind of computing team here. And so as a computing person, would you be willing to share kind of what you found to be the most challenging part of putting this tool together? I mean, how hard is it to work with image files? Do you do anything to do like image shrinkage or subsampling to make them smaller to deal with? Or do you take them kind of it's a lot of image data, and I don't know that people are aware of how large images are in terms of just computing space. And so, I was wondering if you would comment on what was the tough parts of this for you when you were building it.
Yeah, thanks for that question because I get to talk about like computer nerd stuff that people don't normally want to hear about. But I think there are two big challenges. The first was the managing all of the software dependencies and trying to cram them into executable files that people were actually able to use, because there's a lot of the Marco model is actually used as a bit of like the programs it's built on are a bit outdated and don't like play nice with other things in the program. So there's a lot of hacking and manipulation of things to get everything to package together. So I spent a lot of time doing that. And in terms of the image data, Polo does things like when you load a run, you're not actually loading that image data, right? You're just Polo is telling you are, but it's really only doing things like only loading the images that you're seeing at one time, because there's just the amount of data that is produced in terms of just file size is huge, and would trying to load all that at once would just crash people's computers. So there is some things happening in the background to minimize the amount of data that's actually stored in memory at any given time.
That's great. I'm going to go with one more question so that we can move into our next talk. This is from Gabby. What type of errors is Marco most likely to make?
I think it's I can't probably give the most informed answer to this question because I'm not one of the authors of the program. But from what I've seen, it's just, if you have a salt crystal that's looks really like a regular crystal, then it might get tripped up by that because it doesn't actually it only looks at the bright field images. That's the only data that is being interpreted by the model.
Right. And I think that's important for people to realize that the Marco algorithm that you've implemented in Polo really was only trained on bright field imagery.
Yeah,
and
exactly.
So the Polo allows viewers to look at the other image modalities, but they are not part of the actual scoring algorithm. So
yeah, exactly. That's correct.
This is wonderful. Thank you so much for sharing kind of your work on this. And if you can stop screen share so that we can load up our next presenter. And if you could.
Hey everyone, can you guys hear me?
Yep, we can hear. And right now we're looking at you need to go to full screen view.
Wait, do you see the full screen view?
Yeah, now I do. Yep.
Okay,
yep.
perfect. All right, so everyone can see the full screen view and everyone can hear me?
Yep, everything's all set. Thank you.
All right, so thank you all so much for coming to this talk, and thank you to the HWI team for setting this talk up. My name is Nuril Benayan. I am a fourth-year graduate student in the John Hunt Lab at Columbia University, and today I'm going to talk to you guys about how to improve crystallization propensity in proteins that hate to crystallize using lysine to arginine substitutions. So, everyone here loves X-ray crystallography, right? And I guess an overview of how, we all crystallize proteins is, you start off with a plasmid that has your protein of interest encoded in it, right? And then you purify it. Super simple, super straightforward. There's 1,000,001 ways to purify proteins. Most people don't have issues with purifying proteins, but then we want to get a protein crystal. Now you can optimize conditions to maximize the chance of getting a protein crystal, but if your protein target does not want to crystallize, I mean, it won't, right? But if you're one of the lucky few who do manage to get protein crystals that are of diffraction quality, right? You shoot an X-ray laser at it, you get a diffraction pattern, and then voila, you get your protein structure. So the goal of this talk is to show you guys how we at the Hunt Lab have developed a method to optimize your protein's properties to help them crystallize efficiently. So to do that, I want to start by looking at, what exactly is in a protein crystal, right? So bringing the magnifying glass over it, right? What you find is that protein subunits in a protein crystal are, stacked orderly against one another. And if you can see, closely between each protein subunit, they're making crystal packing interfaces, as we call them, between surface residues on each subunit. So, an arginine on one surface of one subunit could be making a hydrogen bond with like a glutamate on the surface of another subunit. And all of these little interactions between different subunits in the crystal help drive crystallization propensity, because the more enthalpically favorable things like hydrogen bonds and salt bridges you can make between proteins in a crystal lattice, the more likely you are to get a protein crystal. So what my lab did was we data mined the PDB for 87,684 crystal structures. And what we did was we simply examined interfaces and asked the question, what is the distribution of amino acids in these crystal packing interfaces? Do proteins that crystallize tend to favor, one amino acid or a set of amino acids on their surface versus others when it comes to driving these crystal packing interfaces? And as it turns out, arginine is a fan favorite of protein crystals in mediating crystal packing interfaces. And, we're like, all right, so if arginines are overrepresented in crystal packing interfaces, if we introduce arginines to the surface of proteins, right, by single point substitutions, then perhaps we would be able to increase the probability of a recalcitrant protein crystallizing, right? So the first question was, well, okay, so we know that arginine likes to make these crystal packing interfaces. We know that it's overrepresented in crystal packing interfaces, but which amino acid can we substitute arginine for or in? And well, lysine, right? Lysine and arginine are very similar in physiochemical properties. They're both basic. Arginine relative to lysine is stickier at the end with that guanidino group. Those 2 or 3 nitrogens at the terminal end, are great candidates for making hydrogen bonds with, oppositely charged residues like aspartate, glutamate, and so on, right? And moreover, since they're both, physiochemically similar in nature, substituting lysines for arginines would most likely preserve wild type function and structure of your native target. So in this spirit, we wanted to find a way. So, if we have a protein sequence and we don't have its structure and we want to substitute arginines instead of lysines, we want to do our best to maintain that protein's native three-dimensional structure and native wild type function, right? So to do that, I've written a program called bulk arch. So it is, we wrote it in Python, right? And what it does is it takes, I said here, it inputs a protein sequence. It actually inputs a multi-alignment file of homologs of your protein target. And it uses specialized sequence analysis and an in-house ranking algorithm to essentially tell the user, all right, here is your protein sequence. Here are all of the lysine sites in your protein sequence. And what it does is it says, all right, first substitute this lysine site. This lysine site, you can put it in arginine and not mess with its three-dimensional structure. Then this is the next favorable lysine site. And it gives this essentially order of lysine sites one can substitute systematically with arginines without perturbing the protein's native three-dimensional structure. So to give you all just a order of operations of how this process works, or first give you guys, I guess, the output of how this program works is What I do traditionally when using this program is I'll take a protein target whose structure is unknown. I'll feed it to the NCBI BLAST and I'll get 5,000 homologs. Then I will use a server like MAFT and align all of the homologs to each other and to the reference, and then input that alignment file into the bulk arch program. Then the bulk arch program works its magic and segregates all homologs into exclusive 10% identity bins. For example, in this particular alignment file for this particular protein target, there were 44 homologs that had 90% or greater identity to the reference sequence. 205 homologs equal to or greater than 80% identity to the reference sequence, so on and so forth. And what it does is it looks at all of the right lysine sites in the reference sequence and asks the question, at these sites across all of these homologs, how does this lysine site behave? Is it flexible? Is it evolutionarily conserved? Right? Am I not allowed to touch it? Does nature enjoy substituting lysine with other amino acids? Specifically, does it enjoy substituting it with arginine? In this particular protein, you can see that site, if you're looking at the bottom right, site 42 looks like it has a high arginine to lysine ratio. So site 42 would be a good candidate to substitute arginines into because, evolution says you can. So the program uses this types of analyses to say, all right, here are these sites you can substitute arginine in for lysine. So the way it works is, let's say you got a protein. In this case, this protein is called My Entire PhD. And all you know about this protein is its primary sequence. You next feed it into BLAST and get a multi-alignment file. You can tell BLAST how many homologs you want. I always go for the max number, which is 5,000. And next you feed it into the program and the program spits out the sites of lysine you should substitute first, and then the sites of lysine you shouldn't touch. So site 125, best one to substitute first, then 6, then 7, so on and so forth. And then what you want to do is take those lysine sites, substitute them with arginine, multiple substitutions. So first substitute 2 lysine sites with arginine. Next, so do the first 2 sites, then do the first 4 recommended sites, then the next 6, so on and so forth. And then grow the proteins, and then run a solubility assay on those proteins. And you want to choose to purify and crystallize the protein that is the most soluble and has the most lysine to arginine substitutions. This will maximize the probability of your arginines making favorable crystal packing contacts and getting a diffraction quality crystal. So we use this method on 2 proteins, one of which already has its 3-dimensional structure available on the PDB and the other one which doesn't. So case study 1, we wanted to see how this method works for a protein that already crystallizes incredibly well. This is a methyltransferase protein, and there's nothing particularly special about it other than it has lysine sites and it crystallizes natively. So these are the lysine sites shown in red, and these are the lysine sites shown by their sites. So I fed the primary sequence into bulk arch, got a multi-alignment file. I fed its primary sequence into NCBI, got a multi-alignment file, fed that into bulk arch, and then bulk arch said, hey, look, these are the lysine sites you do want to touch, and then the red are the sites that you do not want to touch. So we substituted these lysine sites for arginine, and then we purified the protein, and it crystallized. And here is the crystal packing symmetry mates of this target. And then when we ask the question of, okay, so we substituted a bunch of lysines for arginine, are these arginines making crystal packing contacts with neighboring subunits in the crystal lattice? And the answer was, yeah. Here are 2 examples of arginines, our engineered arginines making crystal packing contacts, K8R and K64R. Great. So we can successfully crystallize a already crystallizable protein by substituting lysines for arginines. Our engineered arginines make these crystal packing contacts that are super favorable for maximizing crystallization propensity. And then the final question is, well, how close is this lysine to arginine substituted structure with its native wild type? And when we align or superimpose the two structures, we get an RMSD of 0.32 angstroms. Not bad at all, right? But this protein already crystallizes without any substitutions. So let's do the more interesting case. A protein that does not natively crystallize. So human protein disulfide isomerase domain A does not crystallize. No matter how much you kick, scream, and yell at it, it will not crystallize natively. So we had the primary sequence for this protein. We fed it into NCBI BLAST and then bulk ARG. And by substituting 9 lysine sites with arginine, we were able to make this protein crystallize to a 1.9 angstrom structure, which was great. Okay. And then if we ask the question of, all right, is any of our engineered arginine sites making crystal packing contacts in the subunits? The answer is, well, this is just one example, but yes, K65R makes crystal packing contacts with proteins in neighboring subunits in the crystal lattice. So great. We were able to get a recalcitrant protein to crystallize, and our engineered arginines, again, made crystal packing contacts with neighboring subunits, further indicating that our engineered arginines are indeed helping proteins that don't crystallize. So, another question is, well, all right, so we get this protein that never crystallized before to crystallize. What do these different level of arginine substitutions do to the thermal stability of these proteins? Do these arginine substitutions, make these proteins incredibly unstable? Do they make them more stable? Do they, keep their stability intact? So looking at the right, this is a CD thermal melt plot of different constructs of the protein that does crystallize, which is the bottom, and the protein that does not natively crystallize on top. And the more arginine substitutions we add, the lower the melting temperature of each of the constructs become, but only modestly, which indicates to us that you can add a bunch of lysine to arginine substitutions, add favorable lysine spots, and the worst thing that happens is you only very, very modestly decrease the melting temperature of the protein. And then when we ask the question of, all right, how does the crystallization propensity improve across a crystal screen, which was done at HWI, how do these lysine to arginine substitutions improve the crystallization propensity of these protein targets? So up top, you can tell that after a month, the native structure of HPDIA, there were no crystals, but after a month of the 9K to R substitution construct, there were nine crystal hits that were of diffraction quality, right? And then for the bottom one, the more lysine to arginine substitutions we added, the better, or the more crystals we got. So, and then one last thing to show you guys, when we analyze the crystal packing contacts of our of these structures, and we ask the question, okay, when it comes to when we actually count the number of crystal packing contacts arginines make in the crystal packing files, what do they look like in native versus native versus engineered? Do they increase, and what do they look like relative to lysine? And if you look at the bottom row, when we ask the question of, all right, when we add these mutants, what happens to our arginine crystal packing counts? They go up. And then when we ask the question of, well, what do they look like relative to lysine? They are so much higher. Even in the unengineered structures, arginines make or are responsible for more crystal packing contacts than native lysines. So this is the Hunt Lab in our glory, and we have this software available online for users to use, and it's very simple. All you got to do is put in your multi-alignment file with your protein target in it. You put in your protein ID and you press run, and then you get an output of lysine sites to substitute with arginine. We would love for people to use this. This is the link here, but I just realized that users looking at this can't click on the link. So if you guys want the link or our bioarchive paper, please email me or John Hunt at the following emails, and we would love to send you the paper and the link and the software for you guys to use to help crystallize your own recalcitrant proteins. And with that, I open the floor to questions, and I thank you all for hearing me talk. Thank you.
Thank you. That was fantastic to hear this work. And it's super exciting for those of us working in crystallography to know that these tools are available. And we have a number of questions in the chat. So I'm going to jump in with several of these, starting with Gabby's. Do you think the systematic lysine to arginine mutation will have an effect on protein expression / solubility in any predictable way?
Very, very good question. So what we've seen, so there was a paper that actually substituted a bunch of lysines, not with the same methodology we used, but there was a paper, I can't remember the last author's name. They used GFP, and what they did was they substituted they substituted all surface exposed lysines to arginine. And what they found was that protein expression in the substituted construct went downhill, not precipitously, but significantly relative to wild type. In at least these two constructs, when we've measured protein expression, our protein expression has been great, and solubility, I wouldn't say took a hit. It was somewhat different in one construct. We had that we still need more data on. When we added more arginines to it, that particular construct's solubility went down. So to answer your question, it looks like there was a limit of the number of lysine to arginine substitutions you can add until you start getting weird expression / solubility results. But it is absolutely the case that you can add some number of lysine to arginine substitutions without perturbing expression and solubility.
Great, thanks for that follow-up. I have some other questions in the chat. From Anwar, they point out that they have seen a paper using arginine to asparagine for protein solubilization, and that also works. And they follow up with, what do you think would happen if you used arginine in the crystallization buffer instead of replacing your lysine with arginine?
That is a great question. And to be quite frank, Sinatra, with you, I have no idea. I guess the best way to answer that question would be to actually try it out. But that's actually a really good question. I think that should be a question that ought to be experimentally answered. To be honest, I have no idea, just because crystallization is such a hit and miss thing that right, that you could do everything right and still lose. But I think that would be an interesting experimental venture. Absolutely.
Yeah, this is great. This is one of the nice things about these kind of community discussions, is that we all get great ideas of new things to try. Eric Uckel asks, do you think it's possible to seed wild type samples with your K to R mutant seed crystals to get a quote unquote wild type structure?
Oh, that's such a good question. Oh my God. I think that would depend on, if somehow you a priori knew that your protein target was like on the fence about crystallizing, right? I think if your protein target in and of itself natively is just on the fence about crystallizing, and it just needs, 1 or 2 arginine substitutions to just push it over the fence and make it crystallize, I think absolutely, right? But if you have a protein that's just super recalcitrant, and you knew a priori that you would need a lot of arginine substitutions to make it crystallize, I think seeding it with wild type may diminish its crystallization propensity to a limit, only because if you are introducing favorable conditions for the protein to crystallize, and then you introduce the protein that doesn't like to crystallize into it, I think that'll make it difficult. So to answer your question, yes, yes. And if your protein is only like, on the fence about crystallizing, but if it's super recalcitrant, I'd still try it for fun, right? I could be totally wrong. But I think it may do more harm than good, but you wouldn't know until you tried. Again, great point. Oh my God. These are great questions.
And so I will take the liberty of asking my own question to wrap this up. So in the case where you worked, in your test case where you worked with the protein, where you had a structure that is, perfectly crystallizable, but you also introduced the transitions, the mutations, were the diffraction, I understand those are different proteins now, but were the diffraction sort of levels about the same for the substituted case versus the wild type case?
So I didn't run the diffraction studies or the structure refinement, but from
Okay.
the person who did, Dr. Farhad Furuhar, he said that they were of similar levels.
Okay.
So when it came to comparing, because the native structure has arginine too, right? And we did ask the question of, okay, in the native structure, what's arginine doing, right? And what's lysine doing? And then what's it doing when we substitute all of these arginines and lysines? What we found was that arginines in the native structure, like if you look at an arginine site we didn't substitute and ask that question of what does it do natively and what does it do in the engineered construct, it makes more crystal packing contacts on average in the engineered construct versus the native construct. And I think that's just because the protein would in the engineered construct would reorientate itself such that the arginines just maximize the number of contacts they would make. And I think just natively that follows that if you can make a crystal packing contact, you will if you're in a good environment to do so. As opposed to the diffraction quality, from what I understand, it was the same, but I would need to confirm that.
Okay, that's great. Thank you for a wonderful talk. And I'm going to have you stop screen sharing so that we can get our next speaker loaded up, which is our very own Gabby. And if you can share your screen.
All right.
Okay.
All right. Can you guys see that?
Yep, I can see it
And
and we
hear
can
me?
hear you. Yep, everything.
Can you see my mouse?
Yeah, I could see the cursor moving around.
Okay, perfect. All right. Well, first of all, I wanted to say thank you so much to all of the speakers today. It's been excellent and I've learned a lot, and I'm so excited about our user base and kind of bringing this meeting back in a few years. Today, I just want to tell you a little bit about what's on the horizon for the HTX Center, different developments and pipelines that we're working on. Shown on this slide is actually a photo from Delaware Park in Buffalo. So you're probably very familiar with Buffalo being on the news for all of our horrible weather. But it does actually get quite nice here in the summer too. So I hope at some point all of you can visit Buffalo. But to start off, I just want to go over briefly what I'm going to discuss today. So first, I'll just give a brief introduction to our capabilities and resources. Sarah's already kind of talked about this quite a bit, so this will be brief. I want to go into a bit more detail about our Generation 21 soluble screen because it's been quite a lift to kind of get this sorted, but we feel like we have a really robust system now to generate this screen. And I wanted to give our users just a little bit more insight about what goes into the screen. And then just a few operational notes on Marco Polo. Ethan, thank you so much for your great talk. These are really just going to be, a few notes for our users. I'd like to introduce our new Meritorious Users program and talk a little bit about a highly requested service at the High Throughput Center, which is optimization, and how we're planning to implement this as a fee-for-service model and our platforms and setups for optimization. and then just talk about kind of our vision for the center for the next 5 years and beyond. And of course, I want to plug again, please do fill out our survey, especially in light of some of the things that I'm going to be talking about, because we really do want to gear some of our programs and different developments that we're working on towards the needs of our users. So that link should be posted in the chat, and we would really just love to have your input. So first of all, I just wanted to start broad in case we have any aspiring structural biologists with us today. And I just wanted to briefly compare some of the structural methods that are well established, like X-ray crystallography, single particle cryo-EM, and NMR, along with developing methods like micro-ED that has been discussed a little bit today. So for many of our users at this point, several different methods could be applicable to their system. And honestly, many people find themselves sort of integrating different techniques for technical reasons, or just because each of these techniques have different strengths and weaknesses. But we are finding that X-ray crystallography is still the most commonly used and sought after technique. So useful X-ray crystallography data for macromolecules usually provides a resolution of about 1.7 to 3-ish angstroms. So we can crystallize both proteins and small molecules, as well as other macromolecules like nucleic acids. And the upper size limit in crystallography is largely dictated by whether or not your protein or macromolecular assembly will crystallize. With larger sample sizes, usually comes complexity and sometimes motion between different protein domains, and motion in disordered regions that can prevent formation of the crystal lattice. So it's kind of rare that we'll see, extremely large biological macromolecules that are able to crystallize. So with this, single particle cryo-EM is becoming more popular as more universities get their microscopes online. And this technique usually has a lower size limit right now of about 100 kilodaltons for proteins. Although those working in single particle cryo-EM methods development are pushing this technique to lower and lower sized molecules, as well as pushing the resolution for these samples. NMR is mainly limited by the size of your macromolecule. It typically isn't used for proteins that are much larger than about 25 kD. And finally, micro-ED is becoming more popular, so microelectron diffraction. And as Sarah mentioned earlier, this is a technique where microcrystals are probed with the electron beam from your electron microscope. And this technique is particularly useful in systems where large single crystals are not available. And the upper limit for micro-ED in terms of molecular size tends to be actually dictated by the size of the unit cell that can be accommodated in the tiny size of the microcrystal. So if you're interested in either X-ray crystallography or pursuing micro-ED, of course, the key to success is actually producing crystals. And crystallization requires the formation of an ordered lattice, as we just heard really eloquently from Nouriel. And this can be challenging or impossible with proteins with disordered regions or inherent flexibility. So I'm sure at this point, most of the biochemists' workflow now is to, part of their workflow is to run their protein of interest through AlphaFold. And this can be really useful in combination with disorder prediction softwares to kind of understand a little bit more about your protein of interest and where you expect it to have folded and ordered structured regions versus disordered regions. And in terms of construct design, it's important for crystallography to kind of understand what the landscape of the structure of your protein might look like, because you might want to make some efforts to trim disordered regions to promote formation of this solid crystal lattice and prevent, these flexible regions that will be invisible. So I personally like to run my sequences through a few different softwares and compare between them to see what the kind of consensus is, and then develop my construct design based on this. We've also had people today talk about different crystallization chaperones. So for example, affinity tags like MPB or MVP, excuse me, can be used as crystallization chaperones. So it can be hard to predict sometimes, but one of the main things that I like to do when I begin a structural project is to really work on trimming those disordered regions away and making sure that these regions don't impact the protein activity that you're interested in studying. And this can really sort of help you get the ground running with producing diffraction quality crystals. Next, you really definitely want a highly pure protein sample for crystallization. Even small amounts of contamination can interfere with lattice formation or even crystallize instead of your protein. And so, for example, during the COVID-19 pandemic, we at HWI were trying to crystallize a protein involved in COVID-19. And what turned out to crystallize was actually lysozyme that was co-purified with our protein of interest. And so even small amounts of contaminant can really affect the crystallization experiment. And then our best behaved proteins that we see at the center are homogeneous. And so typically our users will assay this by gel filtration or dynamic light scattering to assess what sort of protein populations they have in the tube. It's also very important to make sure your protein is in a stabilizing buffer when you send it to us. So for example, I used to work with a lot of nuclear proteins and they tended to prefer a higher salt buffer. And that leads me to my next point, which is just that ultimately you're the expert in the system. And so for a lot of these experiments, we look to you as the user to sort of understand what your protein might need for crystallization. And so what I mean by that is you kind of want to understand what protein populations you have when you purify your protein. Does your protein need a cofactor in order to crystallize? Does it show temperature dependent sort of behavior and activity that can kind of give us a clue into what temperature we should be incubating your crystallization plates at? These are some questions that you don't necessarily need to know the answers to, but the more information that you have about your system and your protein, the more beneficial it can be in the process of trying to get a crystal. So to explain a bit more about our pipeline, I just pulled a figure from our newest Jove article, which we hope will really help our users understand what happens to their sample once it gets to the crystallization center. So the first thing, we make our 1536 screening plates fresh every month. And the oil that we use, as we've mentioned before, is paraffin oil. And this is because of its high viscosity, because we do image these plates upside down so that we can get a really good focus on the bottom of the well where that drop is located. So after we put the oil into the plates, we actually plate our cocktails, which you can kind of see in this diagram as this little droplet on the bottom of the well. And we always take a cocktail only image of the plates directly before setup, as Sarah said, as a negative control, so we can understand the state of the well before we add your protein. So for example, some of the things that we've seen in the past are salt crystals in the cocktails. We've also seen debris, so like maybe some dust in the cocktails. And every now and then we, despite our best efforts, we do end up with empty wells due to a missed delivery by our robots. Although this is something that we're always looking to try and improve going forward. So you send us your sample, and you can send your sample either frozen on dry ice, or you can send it to us on wet ice. And then we will use our robotic liquid handlers to add sample to each well in these prepared plates. So switching to the Mosquito, actually, for the plate setup here, we used to use a plate mate, and we used to require 500 microliters from our users in order to be able to kind of faithfully set this up and ensure that every single well was seeded with protein. But now that we've swapped to the Mosquito, we are only going to request about 375 microliters of protein from each user. And so we really hope that this will be something that will lower the activation barrier to kind of shipping your sample to us, save you money, save you time. This is what we're all about at the Crystallization Center. So after setup, we do incubate your protein for the course of 6 weeks with weekly imaging. All the images are collected and transferred to our users via an FTP system that is also integrated into the Marco Polo software, like Ethan told us earlier. And we do highly recommend that our users make a first pass through their crystal images using Marco Polo. And this can help you kind of categorize your images into 4 categories, which are clear, precipitated, crystal, and other. And this really dramatically reduces the number of images that you need to analyze and the amount of time that you need to spend doing this. And so I posted this in the chat earlier, but it takes me about a half an hour to get through a 1536 plate if I need to look at every single well. But if I only need to look at the crystal subset identified by Marco, this can take me as few as about 10 minutes to get through a plate and identify my crystals. And so this is a really useful tool, especially because you do wind up with about 14,000 images from a single experiment in the HTX Center. And I wanted to also mention, this is an image that Sarah showed earlier, but this is our 1536 plate with the penny on top for scale. These wells are incredibly small. And one of the questions that we often get from our users is whether or not we can harvest protein crystals from these wells. And our kind of long and short answer is, you know, it may be possible using capillary methods, and we do have users that have harvested crystals from these plates. This is something that we're interested in looking into in the future, whether we can harvest these crystals or do in situ diffraction experiments to get useful data from these crystals. But just note that we can send these plates back to you, but it's going to be extremely challenging if you're trying to loop them in the conventional way to be able to do so. So every plate at the HTX Center, whatever experiment you're kind of sending us, it's going to receive multimodal imaging. And so these are the UV TPF images that Ethan and Sarah discussed earlier, as well as SHG, or second harmonic generation images. UV TPF identifies aromatic residue containing substances, so proteins, and then SHG images identify chiral crystals. And so using these 2 methods along with our bright field imaging, we can help to verify protein crystals, as well as detect submicron crystals that we may not be able to see with the naked eye. And again, here's the calculation. You get 9 imaging sets from your experiment, which is a little under 14,000 images. So lots and lots of data. So I just wanted to stop for a second and kind of look at a few wells and just ask, you know, do we think any of these wells contain protein crystals? Some of them are a bit more obvious. I think we can say like, yes, that's definitely a crystal. There's probably crystals here, but a few of them are a bit more ambiguous. And so, you know, what can we do if the bright field images are ambiguous? And this is really when the power of the SHG and UV TPF imaging modalities come into the picture. And so shown to the left is our UV TPF, and this has a more diffuse signal, but aromatic residues will fluoresce when exposed to frequency doubled photons. So the incident laser beam is kind of this short beam that can produce these frequency doubled photons. And when it encounters the electron system of the aromatic residue, you'll see fluorescence. And I just want to note that this is not specific for crystals per se. I have seen UV TPF in sort of diffuse proteins in solution, as well as phase separated proteins. You'll also see a UV TPF signal from protein precipitates. So wherever there's protein, you will see this UV TPF signal. And so that's why it's useful to also have SHG, or second harmonic generation. And you can see that this has an extremely high contrast. And so it's very easy to identify regions of your image that are producing SHG signal. And how this works is that the incident beam encounters a chiral crystal, which causes doubling of the frequency of these photons. And if we remember from this equation that we probably all encountered in physics class, the frequency is inversely proportional to wavelength here. And so if you're doubling the frequency of these photons, you actually have the wavelength. And so we observe this 532 nanometer SHG signal. All right, so going back to this, I thought it would be kind of fun to do this as an interactive little poll here, where we could all kind of test our ability to identify protein crystals by eye. And so I am going to try and share this poll with you guys. Is it a protein crystal? Okay, hopefully you can see this. And essentially how this is set up is there are 7 different wells. I would love if you guys could score them: protein crystal, clear, precipitate, or other for each of these, and we could kind of see what the consensus is. All right, so this first one, protein crystal, is very popular. Once I get about like 20 answers in, I'll start going through. Okay. We're kind of between protein crystal and precipitate for the first one. All right, I'll go ahead and start kind of going through these as people get their answers in. Okay, for this first one, so we see the bright field image here, and when I show you the SHG and UV TPF, hopefully we'll be able to identify what these are. All right, so in our SHG signal, you can definitely see very clearly there is crystalline substances, and they seem to correlate in the bright field images with these sort of large, what look to be like crystals. So I think these are crystals. And then if you look in the UV TPF, they are lighting up. So indeed, they are protein crystals. So everyone who answered protein crystal, pat yourself on the back. All right, so for the second one, we have about 77% of those who answered saying it was precipitate. And to my naked eye, indeed, this looks like precipitate. Let's see what the UV TPF and the SHG look like. So actually, there's some crystals hiding underneath the precipitate, as you can see, clustered around the bottom of the well here. And then they are also lighting up in UV, as you can see here. Typically, our UV is a bit lighter and more of a diffuse signal than our SHG. But it seems like many of the crystals that are lighting up with SHG are also lighting up with UV TPF. And this could also be, like I mentioned earlier, a focus issue where the focus plane for the UV TPF is a little bit off of the maximum of the signal, because it images essentially 3 different Z slices through the well. All right, well, number 3, everyone says protein crystal, and I'm going to agree with you guys. That is a bunch of beautiful protein crystals. They look gorgeous on the SHG and, you know, so beautiful on the UV. Love that. All right, well, number 4. All right, we're about half and half protein crystal and precipitate. Yeah, it would be really tough for me to identify which one this is, but with the magic of SHG and UV TPF, you can see that these are microcrystals or little crystal clusters. All right. And well, number 5, again, we're like split between protein crystal and precipitate. Some say other. Let's take a look. And also protein crystals. So these appear to be these kind of microcrystals as well. All right. Well, number 6, some say protein crystal. Okay. Sorry, the poll just moved on me. Okay, a lot of you say clear, which is, yeah, definitely what I would think as well. But if you look at the UV and SHG, there's a really diffuse UV signal kind of down near the bottom of this well, but you can see it lighting up on the SHG. So these are just really tiny nanocrystals. And Sarah told me as well that for this user whose crystals these were the crystals that they ended up using for their diffraction experiments after optimization. And I think she said they were the only hit in the plate, so it's exciting. All right, so the last one, well, number 7, 67% say this is a protein crystal. Someone says precipitate. I don't know about that. But then 27% say other. And you guys couldn't be tricked by me, because even though this has a very strong SHG signal, there's no UV signal here. And so this is actually a salt crystal. And so that's the other power of UV TPEF and SHG imaging, is that you can actually distinguish between protein and salt crystals. Another feature that we often see in the SHG for salt crystals is that the signals will have this weird sort of like overexposed tail coming off of the signal. And I think this is just due to the very strong signal put out by these crystals. So thank you all for participating in this. And I hope you can now sort of see the power of using these different imaging modalities. All right, so next I just wanted to go into some details about the Generation 21 soluble screen, because I think, you know, for the majority of people, when they get crystal hits, they look at the condition where they've gotten the hit and maybe not necessarily think about the other conditions where they weren't hitting in, and, you know, what exactly this 1536 plate actually has in it. And so we have several different commercial screens within our 1536 screen, mostly from Hampton, including the Crystal Screen HT, PEG Ion, PEG RX, the Ionic Liquids, Slice pH, Silver Bullets, and Polymers. We have 5 control wells. And so these are things like water. I think there's like TSEP as a control. So these are just, you know, kind of things to monitor the status of your protein while it is going through screening. And then we have about 700 HWI cocktails. And for the first time this year, we were able to synthesize all of our in-house cocktails using our formulator screen builder, which we were so excited about because this gave us a little bit more control over where exactly these cocktails are placed in the 1536 screen. But also our formulator screen builder is really very precise. So we had a lot of confidence, rather than sort of, you know, hand mixing and hand pipetting, that each of these wells has exactly what we have designed to be put in it. So the only difference for Generation 21 is that a few reagents in the polymer screen have changed. And if this is a problem, you need to take it up with Hampton, because they are the ones who changed their polymer screen. So effectively, they added ethylene amine polymers. So this is PEI. You might use this to transfect your cells, but it can also be used as a crystallization polymer additive. And we've added this in our screen at 0.5% and 2%. We've also added 2 more PEG 3350 concentrations. So I think before we had 40% and 15%, and now we additionally have 20 and 25%. And this comes at the cost of the removal of the pentaerythritol ethoxylate condition that used to be in the polymer screen. So if this was your favorite crystallization condition, I'm sure that it can still be ordered, but unfortunately is no longer part of our 1536 screen. So the updates in the screen are pretty dramatic, and everything on the plate has sort of shifted. And so if you're using our 1536 screen in April or beyond, you will want to upload or download the newest version of Marco Polo that Ethan mentioned, or Macroscope J, which will have the new mapping for our 1536 plates. Otherwise, if you get hit crystals and put them into the old versions of these softwares, you will not have accurate data about your crystallization conditions. And we're happy to answer questions about that as well. So the next thing I want to talk about is just a quality control measure that we've recently implemented that I'm pretty proud of in our screening center. And I want to shout out Tiffany Wright and Elizabeth Snell as they're responsible for a lot of this work. So a question that kind of keeps us up at night in the crystallization center for a while has been, how reproducible can crystallization really be? And this is something that Nouriel hit on a little bit, and that there's a lot of inherent stochasticity in crystallization. And you can set up an optimization plate with 96 of the exact same conditions and get a variety of different outcomes. And so we wanted to kind of get a sense of whether or not there was a way to understand the quality of our screen over time and how reproducible we can possibly make it. So to investigate this in our 1536 well plate system, we relied on the crystallographic model protein lysozyme. So I have some fantastic little crystal balls here that we grew in one of our 1536 plates, and this is indeed lysozyme. And our goal here was to determine a quantitative way to determine if things were going wrong in our screen so that we could figure out a way to correct them before it affected our users. And so I like to compare this to the strategy that British coal miners used to use to monitor for the presence of toxic gases in the mines. So these miners would bring caged canaries down into the mines with them, with the rationale that if there were toxic gases present, the canaries would succumb to the gases more quickly than the miners, because they have this sort of unique anatomy that helps them to fly and get to high altitudes, that allows them and requires them to intake more oxygen than us. So theoretically, this early warning would allow the miners enough time to escape. And so sort of in a similar vein of thought, we rationalized that identifying wells that consistently provided an identical outcome over time would provide us with a set of canary wells to monitor the status of our screen over time, and also between generations, as we're now in generation 21 of our screen. And we've changed quite a few things, including the way that we formulate our cocktails. We thought that this was an important step to take to be able to clue us into any sort of systemic issues that we have in our screen. And one thing that we have actually been doing for over a decade is that we will have 2 lysozyme control plates that we set up every single month. And so we have about a decade of old data that we can kind of draw on to see, you know, how consistent is crystallization over time, and can we kind of identify these canary wells? And indeed, we have about 181 canary wells that we can monitor over time, which is about 12% of the overall screen. And importantly, these wells are spread out all across the plate. And so theoretically, if there was something systematically wrong with our screen, we should be able to not only identify that there's something wrong, but also to go back to the plate and kind of figure out exactly what might be happening. So one tool that has been invaluable in this process is Marco Polo, and this rapidly provides the outcomes list in the CSV format. So this allows us to kind of quickly isolate and compare our canary wells over time. And for the ambiguous wells that Marco can't necessarily call or shows a discrepancy, we do a direct comparison and a visual check just to make sure that Marco got it right. One thing that I experience often with Marco is that it will see sort of the shadow from the edge of a drop, and it will identify this as crystal, I think, because it looks similar to the edge of a very sharp crystal. So the last thing that I want to mention is, right now we don't necessarily have a statistical cutoff for what we deem a pass versus fail in our screen, but this is something that we're going to be working on with Miranda Lynch, our resident statistician. So we look forward to kind of developing this and figuring out exactly, when does the screen no longer pass our QC analysis. But I'm happy to report that our Generation 21 report card, it scored an A on reproducibility of about 96.7%. So we're very highly confident that Generation 21 is going to work at least as well as the past generations, if not better. So just a few operational notes for Marco Polo. Thank you so much to Ethan again for the talk. I just wanted to mention again, there is a patch to be able to download Polo with macOS Catalina to Ventura. This is only for the Intel chip Macs, however, unfortunately, and I think that we will probably be releasing this patch along with the new versions of Polo that have the updated cocktail list for users to download. So currently, Marco Polo is not functional with the Mac M1 chip. And I know that this is a huge bummer because a lot of structural biologists love using Macs, and the M1 chip are the newest versions. But we are looking into options to be able to make this software compatible with this processor. And if you have any experience adapting softwares for Mac M1 chip, or someone who does and would like to help, we'd be very interested in having that conversation. And the last thing I want to mention here is that our file transfer program is being blocked in many cases by university firewalls. And so if you try to log into your FTP and you can't for some reason, one of the reasons may be that your university for users in the past is to try and access the FTP using, maybe your personal Wi-Fi or a coffee shop Wi-Fi, because we unfortunately are not able to surpass these firewalls. You can also reach out to us directly and we will find a way to get you your data. If that has to be Dropbox or if we have to send you a flash drive, we'll do it. So please just let us know if you're having trouble accessing your data. The next thing that I'm so excited to announce is our Meritorious Users program that we're launching for the summer. And this is subsidized high throughput screening. So typically for academic users, our high throughput screen is $400 per plate. We're offering this at only $100 per plate for qualified users. And the qualifications here, if you work at a PUI, if you work at an MSI, or if you're in an under-resourced lab, the definition of which is right under here, you qualify for this $100 per plate, and you can send up to 3 unique samples at $100 per plate. And so you get all of the same things that any academic user would get with our high throughput screen, including expertise by our team. And we just hope that this can kind of lower the barrier to access for our high throughput screening resources, because we do have a lot of fantastic instrumentation and the power of this screen is incredible. And we really hope that this will help people be able to achieve their crystallization goals, or people who never considered doing crystallization before will reconsider because of this option. So please ask questions about this as well. We're very excited to be able to announce this. The application is using this QR code here. I believe there's also a link on the website to the application. It's very short. I think we designed it to take less than a half an hour to fill out. The application is due on May 31st, and you will have your decision by June 30th. So we're very excited to have some meritorious users for the summer. And finally, I kind of wanted to talk a little bit about our in-house optimization and scaling, because this is something that our users have requested for quite a long time, being able to scale the hits from their 1536 screen as well as optimize. And so we are working on a fee-for-service optimization pipeline, and we hope to have this running in early 2024 for access by our users. So here's an example of something that we recently optimized in the High Throughput Center. And this is something that Sarah's academic group is actually working on. And so as she mentioned, Sarah's been really interested in getting microcrystals, and that's one of the things we optimize for. So you can see effectively two different morphologies of these crystals here. We optimize them for micro ED. Up here, you can see very, very small crystals, more visible on the UV TPF and SHG signals. And we also optimize these same conditions for single crystal diffraction experiments at the synchrotron. So we were very excited to be able to do that. And we feel like our pipeline is becoming more robust as we get more practice. We can custom generate formulator grid-based screens for you, customize different drop sizes. There is an optional kind of harvesting and data collection. This is something we kind of need to have a conversation about. You know, with any of our optimization users, that would be part of it. Part of that is accessing, you know, the team of HTX employees and being able to draw on our expertise. So a few formats, we can do sitting drop, hanging drop, and micro batch, just to kind of drive home the idea of optimization and different formats here. This was a hit from a user in our 1536 screen. In vapor diffusion, this could not be optimized, unfortunately, using the sitting drop technique. But as Crystal was kind of alluding to earlier, microbatch was able to reproduce these crystals. So there definitely is utility in trying different formats when you're optimizing. We're also trying to gauge interest in experiments like LCP crystallization, soaking, and co-crystallization. So more ideas to put into the survey if you are interested in these kind of experiments. So for the next 5 years of the High Throughput Center, we're really interested in evaluating the success of our current 1536 cocktails and refining them to better suit the samples that our users are sending us and the most recent sort of information from the PDB as far as crystallization cocktails are concerned. We're going to be developing pipelines to go crystals for specialized applications. So these applications might be micro ED and serial crystallography. We would love to identify either a robust harvesting protocol or a good collaboration with the synchrotron for in situ data collection on some of our crystals. This is kind of a pet project that I would love to personally work on, is a specialized nucleic acid screen. So if this is something that you would like to see from the HTX Center, we would love to hear from you. And then I would also like to announce in 2024 that we will be conducting an on-site training workshop. So we'd love to know, what would your trainees like to get hands-on experience with? Sarah just popped up a workshop poll for us. So we're really excited to hear about that and plan that, and make sure you all have the resources to make this a valuable workshop for your trainees. And I think that's everything that I wanted to say. So again, I just want to thank the HTX Center team, Elizabeth Snell. She's been one of our mainstays in the HTX Center, and she works on pretty much everything. Miranda Lynch, she's our math lady. And Sarah, our fearless leader. She's, you know, so great at writing grants and getting us the resources we need and making sure that the users of the HTX Center are really getting value for, what they put in. And Tiffany, thank you for everything you do to keep this operational every day. So I'd also like to acknowledge our national resource funding, and please be sure to cite this grant whenever you are publishing results that depended on data from the Crystallization Center. And instead of taking questions, I would love to have you guys ask questions in the panel discussion that is coming up next. So for this format, you can either unmute yourself to ask a question or pop your question in the chat. So I think we'll just immediately transition into that.
Great, thanks so much, Gabby, for sharing all the excitement that we've got going on. And I see that people are still weighing in on the poll, so we'll leave that up for a little bit. I would personally like to thank all of our speakers for contributing such wonderful stories from their work, and helping us all kind of learn some new things and be excited about our crystallography. So with that, though, we're going to actually try to open this up a bit for a panel discussion. And so our speakers and HTX Center members are going to be our primary panelists, but we want this to be very much a community discussion. So if you have comments and want to jump in on these conversations, please do so. We're interested in not just hearing your questions, but also in hearing your responses to other people's questions, so that we can benefit. As we've seen in the chat, a lot of people have asked great questions that have pointed our speakers towards some new ideas for approaching their own work. This is the beauty of working in a research community like the structural biology community. And so, we're going to have some questions to kind of get us started, but please don't necessarily feel like the only way you can ask a question is to put it into chat now. We really want to open this up for everyone to participate. So with that, Sarah, do you want to get started with some questions?
I think that there's a couple questions in the chat still that we could start with, actually. So, and if any of our speakers or any of our participants, like Miranda was saying, if any of you want to unmute and put your video on and talk, we're really hoping that this will be a discussion and an interactive thing. We know that there's a lot of expertise in the people who are on this call, and so we want to hear from you as well. So one of the questions is, have you got crystals from large proteins with multiple domains? And if yes, what was the size of the protein? And that's a really great question. And I think my answer is yes, but I don't know the size. So I'm curious if anyone else actually has an answer on that. And if you don't, that's okay. It's something we could look up and I could get back to you, Anwar. So.
Oh, Gabby, you first.
This is from my grad work, but I used to crystallize nucleosome complexes, and we would crystallize a nucleosome, which is about, I want to say like it's over 200 kilodaltons, plus a protein about 50 kD. So approaching, yeah.
Big.
So pretty big.
Yeah, I will follow that up with something not necessarily directly relative to our crystallization center, but during the pandemic, we actually had a chance to put together a paper about the contributions of structural biology to the fight against SARS-CoV-2. And there's an interesting graphic that's present in that paper that shows, for the SARS proteins, which ones had been structurally determined and by which methods. And what was really an interesting take home from that, and we plotted it out in terms of molecular weight. And so I think that gives people a sense that, you know, for the very large SARS proteins like the spike, for instance, those were done essentially exclusively with cryo-EM structures, whereas the smaller SARS-CoV-2 proteins were much more amenable for, and there's the graphic right there, in fact, and this is on a log2 scale. And it shows you that there's, you know, a real ability to work with the smaller proteins, and that the larger ones were, at least at the time that this was put out, much more likely to be done using cryo-EM methods. And so there's definitely some difficulties in working with very, very large complexes and multi-domain proteins in the larger sizes for crystallography cell. So that's a great question.
And, you know, just to jump in here on this particular slide as well, I think that the really, at the time that we put this review out, I think there were a little over 1,000 structures at that point. And I think 4 of them had been done by NMR. So we really just plotted the X-ray structures in purple and the cryo-EM structures. And you can see that there's a lot of area of overlap in those structures. And so again, I think that this is where kind of using multiple techniques and using kind of whatever you need and whatever you can use for the structures that you're trying to solve is going to be really one of the really important things. So I'll stop sharing that. And let's see. And I think Eric had another question that I think, Miranda, you answered, but I want to bring this out to the whole group, that what do you do if you don't have tryptophan residues in your protein? And does UV TPF detect tyrosine as well? So the answer is yes. If you don't have any tryptophan residues in your sample, you can still, if you have tyrosine, if you have phenylalanine, if you've got anything that's got some aromaticity, you will often be able to see some UV signal. One of the things that we will often have to do is actually go into what we say is the Z slices to actually be able to look a little bit more closely at whether we're seeing any signal, because those options just have a less signal intensity, and UV TPF is already a little bit diffuse in terms of what exactly you can see. And so, but we have definitely seen crystals, and crystals have been optimized that have no tryptophan residues. And so that is a definite thing that can be done. And I will say that there are other techniques that are out there in terms of things that can be added to kind of the crystals to essentially help light them up in these techniques. And we are pursuing some of those things as well in terms of thinking about being able to add those things. Of course, anytime you add something to a crystal, you have the potential for disrupting your lattice and then disrupting your crystal. But at least to be able to identify whether crystals are forming, we're working on that more. So, okay. So, are there? Thanks, Eric. Are there any other questions from kind of the audience? We have gotten a couple of questions that we can definitely address. And one of the questions from the earlier part of the panel was, do you take on proteins and projects where some crystallization conditions are known, but diffract to low resolution to further optimize them or find new conditions? And the answer to that right now is not quite yet for most of our non-proprietary users. We are working on it, though. It's part of what we would like to really be able to offer, as well as being able to think about how to offer even a smaller amount of screens. 1536 is a lot of screens, and that's really great as long as you have enough protein and sample to put into the plate. But if you're sample limited, which many of us are, then suddenly that's a lot. And so we are working on not just figuring out ways to kind of set up optimization, but to tailor screening for people as well. That's a little bit down the road though, as we're building up all of these pipelines. Let's see. We do have, again, some other questions that have kind of come in and that we have. And so I'm gonna ask the panel to weigh in. How does protein concentration affect crystallization?
Okay, so it's typically what I tell people if they haven't tried to crystallize their protein before and they're looking for a starting protein concentration is I like to recommend that they concentrate their protein to the highest concentration where their protein seems to be pretty stable and happy. So if you start seeing things like aggregation on your gel filtration columns, if you start seeing weird precipitates while you're concentrating, you've gone too far. Those would be my suggestions.
Eric, do you have a?
Yeah, I mean, of course it makes a difference, right? I mean, you typically kind of have a range, but it's a huge range between different proteins. And I guess in my experience, I aim for 10 mgs per mil to start, just because you have no idea, right? And then if you look at that screen and you've got just loads of wells that are dense precipitate, you're probably too high. If you got loads of wells that are clear drops, you're probably too low. And then that's also a thing that I optimize after you do get initial hits, right? So I'll always change the concentrate, either the protein concentration or the ratio of protein to precipitant solution as part of the optimization procedure, because it's really hard to estimate. And even going from the screen to your, and I use microbatch too, just because like it more directly translates, I feel like, from the screen almost every time. But even then, the bigger drops and the different surface area to volume and things can change stuff. So, but yeah, it's a big range, and knowing where to start is hard, right? So you just kind of like Gabby said, I think the best thing is how much can you get that's feasible, and then start there.
That's great. We have a comment from Anwar, which I am going to kind of give a big thumbs up to also use the pre-crystallization screen or those components from Hampton Research. And that's a good way, especially again, if you're resource limited, sample limited, do a quick screen with a couple microliters of your sample to see if the concentration range is in a good range before you send to us. Because then you know, you're much more likely to get good crystal hits from the screening experiment. So absolutely, Anwar. Okay. You know, one of the things that came up again in the earlier session was a question about, you know, what's the best approach to replicating and scaling crystals generated using the high throughput screen? And I kind of wanted to return to that. And, you know, Eric, you just mentioned that you use mostly microbatch. And, you know, before I came to HWI, I had not really used microbatch. So it was, I have become a convert. And so I wanted to see if anyone else wanted to speak to that and talk about what they find to be the best way to scale up. Because I think this is a problem regardless of whether using a high throughput screen or just a normal kind of screen, a low throughput screen, to be able to optimize from there.
And I'd like to add to that, you know, how when people are getting results back from their crystallization screens and they've got multiple hits, how do you kind of decide which or how many of them you're going to try to optimize? These are all things that we're thinking about and developing ideas, but we'd love to know kind of what the community.
So I guess I can start on this one. everyone kind of has their different approach for how they like to do optimization. But how I like to start after I have a hit, or say like 5 hits from a screen, is look at the conditions and kind of compare between them. Like, do I see any commonalities? Is there something that keeps showing up in these conditions that I should incorporate into my optimization? This is something, as well as I think one pitfall that people, including myself, have kind of fallen into is going too narrow with their optimizations too early. And so I think once you have a hit, you kind of want to have not an insanely broad grid screen, but you definitely want to be sampling, not just like 10 to 20% peg. Maybe I would go like 8% peg to like 30% peg. Like, you want to kind of fiddle with these conditions more than you necessarily would rationalize just from looking at the conditions. I also think, things like swapping out buffers at identical pHs can have more of an impact than you think it will. Changing salts, definitely. Recently, I've seen some results of optimization using these broad peg smears. And I think this is kind of an interesting way to sample a bunch of different pegs at the same time. So there's a lot that kind of goes into it. And part of it too is thinking, how much protein do I have to work with? Do I have the reagents to be able to set up these optimizations and things like that?
Yeah, and I'm going to add one other thing. So I think microbatch actually ends up being almost always more reproducible than vapor diffusion, and I think a lot of us are used to vapor diffusion, and but microbatch can really help. So we do have a protocol for converting, and we're always happy to share that. It should be posted on the website, but I'll make sure that that's the case. And then one of the nice things about microbatch is that you can often harvest directly out of those wells, and the oil itself can be used as the cryoprotectant. And so that can be a really helpful thing, to not have to necessarily screen through potential cryos. It doesn't always work, but it's certainly something to try. And then finally, I am a big advocate of seeding. I think that often, if you can get things to seed with, even if they don't have great morphology in the optimization, that you can end up moving along essentially the kind of use those seeds as nucleation sites to generate better morphology crystals. And so those are additional things that I would add to this. So I think that there was a question from Manas. Is there any method or software that can detect the actual concentration of protein? And I'm going to say I'm not aware of anything.
I'm not aware of, that's an interesting question, and perhaps reflects, something that it'd be nice if there could be development along those lines. But I've seen software to help out with other kind of aspects of this, but in terms of the concentration, I'm not aware of that. So that's a really interesting idea.
Yeah.
I have to imagine that you'd have to sort of calculate the phase diagrams,
Yeah.
and there'd be a lot of experimental work that would have to be done in order to do that.
Yeah, I think part of it is a lack of information. Like machine learning and AI is all really great, but you have to have data that goes into those programs. And if you're like me, you get a crystal and you want to optimize around the conditions that you kind of got. You're not necessarily going back to I got precipitate here, unless you have to, which is something to kind of keep in mind, that if you're not getting any crystallization at all, like into nice big chunky crystals, to think about, okay, well, where are you getting at least some precipitate forming? Because those A could be microcrystals, and B could be essentially just off of the kind of pathway of forming crystals. And so it's a good place to actually start to optimize. I'm going to go to another question, and that is what temperature should I choose to incubate my plates? And I wonder, Gabby, do you want to speak to that?
So in a perfect world where you had unlimited money and unlimited sample, I'd say try it at all 3 temperatures that we offer, 4, 14, and room temperature. But that's not the reality for any of us, even, the richest grant people. So my thought on this is if your protein shows some sort of temperature dependent behavior, this is something that you're really going to want to kind of think about and consider how your protein and how the cocktails might interact at lower temperatures versus room temperature. But if you're just working with a protein that seems to behave similarly at room temperature and in the cold room, I would probably suggest just to incubate your plate at room temperature to begin with. And then during optimization, you can kind of fiddle with temperatures to try and control the growth rate of your crystals.
Yeah, and I see Amar, we usually use 4 and 18 degrees. Thank you. If there's anyone else who has a favorite temperature, this is always something where it's one of the big parameters, but of course you have to have an ability to incubate your plates at that temperature and try to make sure to be able to image the plates at those temperatures. So for our 4-degree plates, we actually image those at 14 degrees. We don't have a 4-degree imager. And so that's another thing to kind of be aware of, that we do move those around. And so there can be slight changes in equilibrium at that point. But and I think one
it
of
also, sorry, Sarah.
No, go ahead.
I was going to say, I think it also makes a bigger difference if you're using like a hanging drop or like a sitting drop versus micro batch due to the evaporation.
Right. I completely agree. Let's see. I'm going to ask one other question here. Again, feel free to weigh in with questions or feedback or anything like that. And this is a question that kind of came up with regards to Eric, your talk, and something that I'm also interested in. Are there any special things that need to be done to kind of work with metals in kind of crystallizing metalloproteins? And so, Eric, I don't know if you want to speak to that or.
Yeah. So, with ours, most of those express with some metal bound. And then zinc is pretty common to pull out anyway. And so we've been kind of fortunate. We don't usually add, what I've seen with zinc especially, but I think this is true of other metals. If you try to saturate your protein and then you go to crystallize, it tends to precipitate. So divalent cations kind of do that anyway. And it's the same as sort of the balance between precipitation and crystallization. Is a little bit's good, probably, and a lot is probably bad. Many things will crystallize in one form and not the other. So we did have this sort of odd experience with the Zerge protein that only the apo would crystallize. And so you can try both ways. Usually initially we will take them as isolated and try to crystallize. Even if it's substoichiometric in metal, because you're basically going to purify out the form that you get in the crystal anyway, and the occupancy is usually pretty good. And then we'll try to remove them. And normally what we do for most of our stuff is we'll dialyze against a chelator at low pH, as low as your protein can tolerate without totally crashing out. And that'll protonate some of your like histidines and things like that, and allow the chelator to snag up that metal. It works pretty well. The other thing I think to think about is if your proteins are his tagged. There's a lot, I think, of evidence to suggest that metals will promote dimerization and potentially precipitation with his tags because they're just going to like stick. They're all going to glom onto it. So whereas a his tag is probably fine in most cases, with metalloproteins, it's kind of, I avoid it for sure. So we just purify with ion exchange and other untagged versions. So yeah, it varies a little bit. I think, again, for our strategy, we just kind of go with however we isolate it. We also try an APO version, and if you must, you can try to add metal to it, but I find that it usually doesn't work. And a lot of times you can go back in and soak later. We've seen that can work too. It can also totally explode your crystals. It'll do that too. It's just hard to tell how big a conformational change you'll see. If it's modest, it usually doesn't disrupt anything too bad, and you end up okay.
Such is the joy of crystallography, though, and everyone who's out here is like, yes, we've all had those dissolving things and exploding things, and so lots of sympathy.
Yeah, you know, and I'm so a number of the projects we work on are metalloprotein projects, and so I am going to concur with Eric on these things. You know, for some of our things we can do apo crystals and then soak metals in, because when we add the metals to the solution, it actually precipitates out. There are other times when we actually just add the metals to the crystallization cocktail, and then essentially add enough metal there so that when it's crystallizing, that we end up getting some good crystals. But that takes a lot of fine tuning to kind of figure those things out. There is a question in the chat from Anwar. What if your protein contains both zinc and magnesium or multiple metals? And the answer, you know, for me is that, yeah, there's that it can be hard to make sure that you've got the right metal in, the physiologically relevant metal in your sample, and B, there's also times when your sample is actually going to be promiscuous in terms of metal binding. And so, you know, we have to think about, well, is that going to impact what the kind of any of the functional things that we're thinking about are happening to that we're using our structures to kind of base our hypotheses on. And so there are methods that can be used to actually identify what metal is in your sample, either by doing anomalous data collection to be able to look at what metals are explicitly in different sites, as well as doing things like energy dispersive X-ray spectroscopy to be able to identify what is in your sample. That can be done on both crystals and solution. And so there's a lot of different things that can be done for this, but it's something to definitely pay attention to. So you know, I think it just adds to the extra little bits of challenge to doing protein crystallography to have to also be working with metals. So let's see if there's any other kind of questions from kind of the audience, but I think we might be wrapping up soon here. I really appreciate everybody's participation. I'm gonna go ahead and put the link in for the survey one more time. And so and you will also probably get it in your email, so I apologize for the spam. But you know, again, feel free to reach out to us if you've got questions, if you've got things that you're trying to crystallize that you're working on and you just want to kind of talk to somebody. We're always happy to help out. And we really appreciate your participating and your presence here in our first ever HTX Center user meeting. We really hope to have, first of all, a workshop sometime next year, and then a in-person meeting, probably 2025 for a user meeting. So again, if you can always check our website. We keep that pretty up to date, although I do need to put the meritorious user application on there. So look for that soon. For those of you who have expressed interest, I'll also email it to you. And then as well, you know, if you're interested in receiving notifications, we have an email list. And so you can sign up to receive notifications for when our screenings are happening, as well as sometimes just news updates about things that are happening in the crystallization center. And so with that, I would like to just say thank you again for attending, and thank you so
and
much
many-
for all of our speakers for participating and you know, giving such great talks. I think it's been really great. And yeah, thank you.
Thanks, Sarah. Bye, everyone. Have a good one.
Thanks, Ethan.
Thank you.
Bye. Thanks, Noriel. Thank you, Eric. Thank you, Gabby. Great talk.